Welcome to the Microbiome Cloud Computing module. In this video, we will talk about common microbiome analysis plots that you will see during the workshop. After obtaining DNA sequencing from a 16S sequencing run, researchers often generate a rarefaction curve as an initial step in data analysis. This curve evaluates the sequencing depth by randomly subsampling reads from the dataset and calculating the number of distinct species observed. In the resulting plot, the x-axis represents the number of reads sampled, while the y-axis indicates the number of species identified. As the sample size increases, the number of detected species typically rises until it reaches a plateau. This plateau indicates that additional sequencing depth is primarily providing more data on already identified species rather than uncovering new ones. By comparing rarefaction curves across different samples, we can assess differences in species richness, such as which samples contain a greater diversity of microbiota taxa prior to further analysis. We can then measure the alpha diversity of each microbiome sample and compare the alpha diversity between healthy, overweight, and obese individuals. The Shannon index is a very common method of assessing this by measuring species richness and species evenness. Shannon diversity asks how many different species are present and what their distributions are. For example, a community with 4 different types of bacteria, each at 25%, will be more diverse than a community with 4 different types of bacteria, with one dominating at 94% and the other 3 bacteria at 2% each. A higher number shown on the y-axis corresponds to higher diversity. In order to visualize differences between microbiome compositions, we could use ordination techniques such as a principal coordinate analysis or non-metric multidimensional scaling, shown in this figure. These methods reduce complex, high-dimensional data into a two-dimensional representation by summarizing the raw data and assigning each sample a position in the plot. Each point represents an individual's microbiota composition, which we can then color code by the amount of protein they ingest. The closer 2 points are to each other, the more similar their microbiota are. Typically, samples with similar characteristics from distinct clusters indicating shared microbiota traits. However, we do not see that in our data based on protein intake. Next, we can dive a bit deeper into the composition of each individual using stack bar plots. These visualize the proportions of bacterial phyla across individual samples. The x-axis shows each sample, while the y-axis represents the relative abundance of bacteria. Each color corresponds to a specific phylum, which may include multiple species. By comparing groups, such as those with different protein intakes, we could attempt to identify variations in phylum abundance between them.